You will hopefully see them in a few months. So the work has been done, obviously, in the entire consortium, but as Data Analysis Center, we actually had a lot of actual analysis role to take. And here are 3 then students in my lab who did a lot of work to hold this paper together for the consortium. Jill Moore now is an assistant professor with her own lab now, and Michael and Henry's here, who is going to talk next. Michael and Henry are MD PhD students, and Michael is a resident now. And Henry also defended going back to med school. So they played quite sizable roles in this effort, so you can see they're prominently featured as co-first authors of this paper. So just a very quick overview as to the data. So I mentioned 4 types of regulatory elements. I just want to use enhancer as an example as to how we define these elements. Because this is enhancers are the largest category of our elements. So typically an enhancer, if you think about it from an epigenetics point of view, an enhancer is a region in the genome where a nucleosome has been kicked off the DNA and make room for binding by transcription factors. Why do I define it in such a specific way? It's because if you look at our DNA, it has to be packaged into chromatin in order for storage and regulation. I mean, our DNA altogether, if you connect 23 pairs of chromosomes, including the sex chromosomes, altogether it's like 2 meters long, and they're just really skinny and long fibers of DNA and heavily charged. So it's very unwieldy, and only when they are packaged around these histone octamers, they can be stored and regulated. Because they are in this kind of chromatin structure, regions that you don't need to use are packaged away, and regions you want to use, such as regulatory elements, are unpackaged, so that you kick off the histone proteins and then make the DNA accessible to regulatory proteins. Okay, so there are actually around 1,600 transcription factors encoded in our human genome. That's quite a bit of them. That's why I show them in different colors here. Experiments can be done using ChIP-seq, and Y here indicates an antibody, and you can raise antibody against each and individual one of these transcription factors, and you can shred the chromatin and pull out the fragments of the chromatin that are bound by a particular transcription factor, for example, the blue one here, and then map the DNA to the genome and find out where this blue transcription factor binds in the genome upon sequencing. And ENCODE has done a lot of these kind of TF ChIP-seq experiments. At some point, it reached maybe 5,000. Just like, okay, greater than 5,000, stop updating them. And also, the same technology can be used to probe histone modifications. So these histone proteins, their N-terminal tails, can be covalently modified in many different ways, in many positions, like phosphorylated, acetylated, methylated, ubiquitinated, in each way you can have, and they demark, they're like little flags in the genome to show you potential functions of that region. And you can also raise antibody against each modification, and then find out where they are in the genome, and then using the same kind of assay, ChIP-seq, and ENCODE has also a lot of these kind of experiments across different histone marks and across different spile samples. Lastly, I want to mention that for open chromatin region, so enhancers are typically regions that are open chromatin because the nucleosome has been dissolved in that spot. You lose one nucleosome there. And these regions can be easily detected by assays such as DNase-seq, which is just an endonuclease DNase. You kind of chew it up a little bit so you don't chew it into bits and pieces, chew it up to fragments. and then you pull out the region that's open and sequence those regions. You're probably more familiar with the attack-seq assay, which is similar to DNase, but instead of using the endonuclease, it uses TN5 enzyme to intercalate transposase into the genome. But that's pretty much the same thing for detecting open chromatin regions. The reason that we use a lot of DNase-seq experiments in ENCODE is because there is one production center by John Stavropoulos at University of Washington in Seattle, and he makes really, really good high-quality DNase experiments. So ENCODE has probably the largest collection of high-quality DNase assays. So we have access to these experiments. So you can tell that being a data analysis center, we actually have access to tens of thousands of experiments, and we were very busy trying to convert them into knowledge, not just reads. So here I'm showing example data for DNase, which shows open chromatin, and then two types of histone marks. Histone 3 lysine 4 trimethylation. This is a mark for active promoters. And K20's histone 3 lysine 27 acetylation. This is a mark for active enhancers. CTCF is the only insulator binding transcription factor in mammals. So this is a transcription factor. So where CTCF binds indicates where insulators are. So here is an example of a gene. SP1 actually encodes a transcription factor. You see exons here, and the beginning of the gene is here. So the beginning of the gene is right next to promoters, as my schematic showed earlier. A promoter is right upstream of the beginning of the gene. Transcription starts side here. And it's typically exemplified by open chromatin regions flanked on two sides of this open chromatin region by characteristic histone marks. So high H3K4ME3 and high K27 acetyl. And then enhancer is usually far away from a transcription start site, also marked by this enhancer histone mark, and also has some transcriptional activities. A lot of enhancers are transcribed. So it also has high K4ME3 and open chromatin. And the insulators, you can see, is also open chromatin and bound by CTCF. So I just want to emphasize that you probably have noticed like all these regions are lined up in the genomic coordinates pretty precisely. It's because it's the same region, but just marked by different features. And that allows us to detect these regions pretty accurately. So, so we aimed to identify a whole bunch of regulatory elements. And we tested different kinds of approaches, and at the end we opted for an approach that's really closely relying on the data we had in hand and taking advantage of the rich data generated by the ENCODE consortium. So we decided that we are going to anchor all these regulatory elements by DNase sites. These are DNase hypersensitive sites. These are the regions that are hypersensitive to DNase cleavage. They indicate open chromatin regions. They usually indicate regulatory elements without specifying which kind of regulatory elements, but it's more like an all-encompassing definition. So you can see that here, every track here is a different biosample, and there are maybe 20 biosamples here, and you see the DNase hypersensitive sites line up pretty straight along the genomic coordinates. Like this is the same SP1 gene I showed earlier as the example. This is the promoter of SP1. And you see that this is the enhancer upstream of SP1. It's open chromatin in some of the cell types, but not open chromatin in some other cell types. And you see these other regions. So if you look across the genome, you can spot these locations that become open in one of the 2,000 DNase experiments. experiments. And we call these regions representative DHSs, RDHSs, and they give us a first inclination as to where the regulatory elements may reside. So for ENCODE phase 3, we defined 2.2 million, this kind of regions, in human and 1.2 million in mouse. The reason we have more of them in humans is just because we have more experimental data in humans than in mouse. And we expect the mouse should be roughly the same, 2.2 million as well. So just to go back to my example, so what do I mean by these regions? We first define these open chromatin regions, and then we combine the definition with additional 2 types of histone marks to define promoters and enhancers, and then CTCF binding to define insulators in at least one biosample. So ENCODE has like 1,000, over 1,000, 1,500 human biosamples. And for phase 3, that paper that came out in that Nature paper I showed earlier, we defined roughly 1 million human candidate cis-regulatory elements. The little c stands for candidate, and because reviewers of that paper insisted that these are epigenetically defined elements without functional assays, and we really shouldn't call them just cis-regulatory elements straight. So we added a candidate in front of the CRE, candidate cis-regulatory elements. And we defined them as promoters, and roughly 35,000 of them for version 3. So they are defined using HG38 genome version, in case you wonder about technical details. Enhancers, I mentioned earlier, we have a lot of enhancers. Some of them are proximal to the transcription start site. Some of them are distal. And then there are 2 other categories. One is possibly novel promoters or poised enhancers, 26,000 of them. And these are boundary elements, insulators that are bound by CTCF. So altogether, 1 million such candidate cis-regulatory elements. Go back to my example of a specific gene. So here I'm giving you an example of HNF4A. This encodes for another transcription factor. This one is specifically functional in hepatocytes. And you can see that for this gene, if we look at the hepatocyte, if you look at all the CCREs here, these are the boxes. The gray ones are the ones that are not marked by these assays in hepatocytes, and the colored boxes are the ones marked by at least one of the assays. So you can see promoters of this gene is marked because this gene functions in hepatocyte. And then this is possibly an enhancer, and that's possibly an isolator. So out of the total of 1 million, usually roughly 100,000 of them would be marked in a particular cell type. And the rest of them are no signal. So if you look at the different cell types in a human body, different subsets of them would be marked. And it's these choose of different subsets in different samples and tissue types that defines the tissue specificity of our regulatory programs. So I actually, yeah. So let me just add one more sentence. So if you don't look at the hepatocyte, I actually had a slide I wanted to include, but I don't see it now. I had a slide that had a neuron, and in a neuron, this gene is off, and then these will show no signal, but it will show neuron-specific sites. So, so that was what we did in ENCODE, and because we had data between human and mouse, we also looked into the evolutionary conservation of these CCREs by comparing between human and mouse, and also comparing across the 100 vertebrate genomes. obviously, here is we're using a phylo-P score, which is a well-known score that you can use to indicate evolutionary conservation across multiple species. As I said, it's 100 vertebrates. And these different types of CCREs show different levels of evolutionary conservation, and we compared between human and mouse. In case you're wondering, I have a rule of thumb of a third rule in life. Usually things are a third, and a third. So between human and mouse, a third of the CCREs are homologous and defined and in both species. A third of it is homologous but not defined as CCREs in both species. And then a third of it is no homology between the species. If you keep that in mind, it might be useful. The reason that we have fewer numbers of them in mouse is because of data, but you see the ratios are roughly the same. So at that time, it was quite intriguing that even though on average CCREs are evolutionarily conserved, then more conserved than randomly chosen genomic regions as indicated by this gray line. There were quite a few cis-regulatory elements that were not conserved. So when after the paper was published, we keep on pondering over this. So are these CCREs that showed low phylo-P scores, are they truly not conserved? Or would they show conservation in more recent evolutionary time? Vertebrate is a very long evolutionary distance. So we were wondering what we can do. So luckily, a faculty member in my program, Eleanor Cole,